Hello everyone, my name is Akshay Rajeev Menon and I'm an undergraduate researcher in Dr. Circe's lab at the University of Texas at Dallas. Today I'll be presenting the results from my experiment. Let's get started, shall we? To start off, the goal for my project was to formulate an acoustic biosensor to detect blood oxygen level. We know that tumor hypoxia is quite common and it renders chemotherapy and radiotherapy ineffective. Therefore, it plays a vital role in developing a treatment plan for the patient. For our biosensor, we decided to use hemoglobin microbubbles. Microbubbles, as you can see from figure 1, are an ultrasound contrast agent. My goal was to use their acoustic response to detect presence of oxygen. For the shell material, we decided to use hemoglobin. This is because we know that hemoglobin can reversibly bind to oxygen, and we hypothesized that the mechanical changes it causes could be used to differentiate oxy from deoxy samples. Now let's take a look at the method of formulation. I first added the hemoglobin along with tryptophan in a solution of 1x PBS with 10% glycerol and 10% ppg. I heated the solution to 50 degrees Celsius to unwrap the protein, as 50 degrees Celsius is the denaturing point of hemoglobin. To generate the microbubbles, I probe-tip-sonicated it while flushing it with PFB gas. Flushing it with PFB gas encapsulated the gas inside the hemoglobin shell. Then I centrifuged it along with a syringe transfer to concentrate the size that was desired. Now let's take a look at the results. In figure 5, plot A, you can see that we had a lot of crosslinks. This is how we got when we first started out. And to reduce the crosslinks, we added tryptophan. This is important because cross-linking increases the size and any particle above 10 micrometers cannot be used for in vivo applications. Then we tested the different absorbance spectra for different concentration of tryptophan. Although higher concentration tryptophan reduces cross-linking, we see that at high concentrations of around 5 mg, we denature the protein to such an extent that it no longer serves its function by binding to oxygen. So we decided to go with 2 mg per mil. Then here's the plot for the size distribution from the multisizer. We can see that the average size of the microbubbles we made were under 4 micrometers. This means that it's safe for in vivo use. To test if we lost the oxygen binding capabilities, we did another absorbance spectra. We can see there is a difference between oxy and deoxy microbubbles. Now let's take a look at the acoustic setup. In the acoustic setup, I set up oxygenated hemoglobin in the acoustic chamber, then proceeded to add the reducing agent, which turns it from oxy to deoxy state. I measured the acoustic response over this time period. Let's take a look at the results. Here we can see the results for the acoustic experiment. First, we see that the dissolved oxygen content in plot A. We see when the addition of the reducing agent dropped the oxygen levels drastically. On plot B, we see the data collected by the chamber. On plot C, we see the acoustic response, known as the stable cavitation dosage for the air-filled hemoglobin microbubbles. We can see that in the plot in the blue line, the addition of the reducing agent caused a decrease in the acoustic response. On plot D, we see the same, but for PFB-filled hemoglobin microbubbles, which was the goal of my experiment. We also see that there is a difference between oxy state before the addition versus deoxy state after addition. In conclusion, we were able to develop an acoustic biosensor that was able to detect the presence of oxygen. We found that addition of tryptophan to the microbubble solution helps reduce the amount of cross-linking. We also saw that the addition of PFB as a gas core helps increase acoustic signal while not compromising the oxygen binding properties. In future, we would like to test the photoacoustic properties of this solution, since hemoglobin itself is known to have an absorbance spectra in the near-infrared region. Thank you. This concludes my presentation. Hope you enjoyed. Have a good day.